Shortly after fertilization, the fertilized egg or zygote begins dividing. By about 4 days after fertilization, a solid ball of cells called a morula forms. The cells continue to multiply, and by day 5, the cells separate into 2 groups, the inner cell mass, which will become the embryo, and an encompassing sac called the trophoblast, which will develop into the placenta. Trophoblast cells secrete fluid, creating a cavity, the blastocele, with the inner cell mass at one end. At this stage, the mammalian embryo is called a blastocyst. The cells of the inner cell mass eventually form 2 layers, the epiblast and the hypoblast. The epiblast is thought to contain the cells that will generate the actual embryo. Critical to the early development of all vertebrate embryos is the process of gastrulation, which occurs between days 13 and 19. Gastrulation results from the movement of cells toward the midline and then forward along the midline, forming a groove called the primitive streak. The end result is the production of the 3 primitive cell layers, the endoderm, the mesoderm, and the ectoderm. The nervous system develops from the outer layer, called the ectoderm. As this cell layer thickens, it becomes a distinct epithelium called the neural plate. Uneven rates of cell division form a groove called the neural groove that will become the midline of the embryo. Ridges of ectoderm continue to bulge on both sides of the midline. The tops of the neural ridges eventually come together, forming the neural tube. The interior of the neural tube becomes the fluid-filled cerebral ventricles of the brain, the central canal of the spinal cord, and the passages that connect them. By about 24 days, the 3 major subdivisions of the brain are discernible. These include the prosencephalon or forebrain, including the telencephalon and diencephalon, the mesencephalon or midbrain, and the rhombencephalon or hindbrain. The progenitor cells of the neural tube are known as neural precursor cells or neural stem cells. This represents the first stage of neural development, neurogenesis, when undifferentiated cells undergo mitotic divisions to produce either new stem cells or neuroblasts that will eventually differentiate into neurons. The cells undergo a stereotyped pattern of cell movements as they progress through the mitotic cycle. Eventually, the dividing precursor cells form a closely packed layer of cells called the ventricular zone. Eventually, some cells leave the ventricular zone, forming a second layer of cells called the marginal zone. Later, an intermediate layer develops as the wall thickens. Cells in the intermediate zone will then begin to differentiate into both neurons and glia. As the number of cells increases during the period of neurogenesis, the newly formed cells must move even greater distances. The massive movement of nerve cells to establish distinctive cell populations represents the second stage of neural development, cell migration. Cells do not move in an aimless, haphazard manner. Cells in the developing cortex move along the surface of a particular type of glial cell called radioglia. Like spokes of a wheel, these radioglia cells extend from the inner to the outer surfaces of the emerging nervous system. The radioglial cells act as guide wires, and the newly formed cells creep along their processes. Once cells reach their appropriate destination, the third stage of neural development begins, differentiation. At this point, the cells begin to use or express particular genes. This allows the cell to acquire the distinctive appearance and functions of neurons characteristic of that particular region. As the process of differentiation proceeds, the fourth stage of neural development begins, process outgrowth. The biggest change in brain cells early in life is the extensive growth of axons and dendrites and the proliferation of synapses. The neural environment also greatly influences nerve cell differentiation. In vertebrates, Young neural cells seem to have the capacity to become many varieties of neurons, and the particular type of neuron that a cell becomes depends on where it happens to be and what its neighboring cells are.